Foodborne illness, or its more common name, food poisoning, is an infection of your intestines, small and large, caused by bacteria. The bacteria that causes the disease is usually found on food that is poorly prepared. The main illness that is associated with foodborne illnesses is gastroenteritis, aka the stomach flu. Gastroenteritis is an inflammation of the stomach and small intestine. It restricts liquids and nutrients from being digested properly, which results in vomiting, diarrhea, and dehydration. It attacks the epithelium, which contains all cells that specialize in digestion and absorption. The bacteria can be spread due to poor food conditions, but it can also be spread through contact with an infected person. One of the most common types of bacterium, Escherichia coli, is spread this way. This is the bacteria that will be discussed in this video. Some quick facts about E. coli is that it is passed through fecal contamination. Feces cannot be digested and they contain the pathogen that causes E. coli. E. coli can cause gastroenteritis and grows ideally around room temperature. It grows exponentially through binary fission and can survive harsh conditions when it is in its spore state. So gastroenteritis begins in the gastrointestinal tract, which contains the stomach and the small intestine. When you eat a meal, the food goes to your stomach and the digestive tract will digest it. However, when you have gastroenteritis, the digestive tract can't digest or absorb nutrients, and your body will want to expel these excess nutrients in your body. The excess nutrients can leave through the esophagus, which will lead to vomiting and nausea, or it can leave the body in the opposite direction and expel itself through the intestines, which leads to diarrhea. There are a number of infectious pathogens that can cause gastroenteritis, such as E. coli. These pathogens enter the system through fecal-oral transmission, so your feces is what doesn't get digested and is excreted as waste. The feces can't contain the pathogen, and once it's in your system, the pathogen will attack the gastrointestinal wall, which covers the entire gastrointestinal tract. And this is where we will be focusing our attention on. Okay, so now we have a zoomed in picture of the gastrointestinal wall. The main focus is the green structure, which is the epithelium, and the epithelium contains all cells that specialize in digestion and absorption. Okay, so let's say we just ate some food and the food contains the pathogen for the bacteria. The pathogens are represented by the small red shapes to the left. The pathogens will then invade the epithelium, so it's going to invade the epithelium cells and the cells are going to die off. So that's represented by the opening in the epithelium. This can lead to dead epithelial cells, which you can see underneath the pathogens. So these cells now cannot digest or absorb any more nutrients that come into your body. Since water isn't being absorbed by the epithelium, there will be an excess amount of water in the undigested food instead of going into your system. So some of the food goes undigested, which leads to vomiting, and the excess amount of H2O causes the waste to come out as diarrhea through the intestines. And since there's also less H2O in your system, you'll be dehydrated as well. So that's kind of an overview of how these symptoms arise from gastroenteritis and the E. coli that enters your body. Okay, so now we're going to move more into depth on how the pathogen attacks the gastrointestinal system. So to begin, the epithelium is covered with microvilli to increase the surface area for absorption. But after the pathogen attacks the epithelium, the microvilli will disappear from the patch of the cell surface that the pathogen and bacteria attacked. And a bacterium will come into closer contact with the intestinal cell surface, which leads to the next step in the infection of the gastrointestinal system. Here we see the pathogen beginning the process of attaching itself to the vulnerable epithelium. The pathogen has two major components on it. Those are the virulence plasmid, which contains the genes for initial binding to epithelia, as well as the genes for intimin. And then timin is the protein that allows the bacterium to bind very closely to the host cell and release the toxin into the body. So the intimin protein will bind the bacterium on the intestinal cell, and the actin will form, which push the two together, forming a pedestal-like structure that allows the bacteria to invade the cell and thus infect it. To understand that, we'll take a closer look. The red cell at the top represents a bacterial cell, and the black cell on the bottom is the intestinal cell from the epithelium. So to invade this cell, the bacteria uses a specialized injector called a type 3 injector system that acts as a syringe to introduce several bacterial proteins into the cell. The green needle-like tube called the ESPA attaches from the bacterium into the intestinal cell. It transports two proteins called ESPB and ESPD that travel through the tube to form an opening in the intestinal membrane to allow things to pass through. The two proteins create an opening by attaching themselves to the cell wall of the intestinal cell as shown here. Now that there's an opening, the bacterium will transport a receptor protein through the tube into the intestinal cell that is called a tear protein. Once the receptor proteins are in the cell, they attach themselves to the intestinal cell membrane, and a portion of them will project themselves to the cell surface and bind to the intimin proteins, which are the blue ball-like structures on the bacteria. And once they do that, the membranes of the intestinal cell and the bacterial wall will be locked together, and this is really where the cell begins to be in trouble. Now the proteins attach to each other, and the cell membranes of the bacteria and the intestinal cell are firmly locked together. So now intestinal cytoskeletal proteins begin to bind to a portion of the tear proteins inside the cell, and the formation of actin filaments forming and building up of the protein begin to take place. Actin filaments build up directly beneath where the bacterium cell is bound to the intestinal cell, and as they lengthen, they begin to push up and form a pedestal formed by the intestinal cell. Okay, so this is the final step, and as you can see, the cell membranes of the bacteria and the intestinal cell are pushed together, causing the intestinal cell to form a pedestal-like structure, which finally allows the symptoms of the infection to commence. And that's how foodborne illnesses invade and infect your body through the bacteria. Now all that's left is to watch an animation to get one last look at how foodborne illnesses affect and invade your body.